able to, the room is small enough if I just play through my computer.
Let me help you with that.
Okay.
And you can also, Bluetooth onto that.
Okay, great.
Which makes it nice and loud.
You need help connecting to it.
That looks good.
Super scrappy. No, that's yours.
I think the audio will be fine from the computer.
Let's make sure that we can at least try to connect to the.
Try to get some of the motion data.
Here.
I've done a bunch of time stacking, but so up here I took how long? Basically took the meeting.
Can you guys hear me okay?
And that gets really fish. Then I stacked a few rounds. And
Right.
managed
I'm really delighted to be here. Thank you so much for the invitation and connecting us. Let me make sure I've got the display going here. Okay, that looks good. All right, so I'll be talking today about canary song and a self-supervised model focused on parsing the syllables of canary song. I come from the University of Oregon, and my lab does some microfabrication, some systems neuroscience, some 3D printing methods development, and some AI for communication. And what that means is, with a very small lab, I do everything in a very mediocre way. Lab meetings are really hard because no one can talk to each other. But some of how we ended up in such an unusual place, I was trained in physics, nonlinear dynamics, really interested in fractals and pattern formation, but I became fascinated as a PhD student in canary song and birdsong generally, and just the signal processing challenge that species offered. Ended up working in birdsong neuroscience for postdoc, but then we needed better methods, better electrodes, better brain implants for the biomedical work that we were doing with birds, essentially trying to understand the interactions between basal ganglia and cortex in motor control. And so we did a lot of development of new techniques, ultra-small electrode arrays and things. And around that time, my mother-in-law was diagnosed with ALS, and I became very motivated in doing something for humans with that, and I took a leave of absence. I ended up starting a couple companies related to brain-computer interfaces in humans. And then about 5 years ago, returned to the University of Oregon, and now I'm back to birdsong behavior. And I actually came back and started focusing in the last year on a problem that I started 25 years ago as a PhD student, which was we should be able to automatically parse these syllables. Like, of course, it's easy for me to do this. It should be easy for a computer to do this. I foundered on that notion 25 years ago, and we restarted again more recently and more successfully. So this is the bird, the species. So I fell in love with that species working with Fernando Nottebohm at Rockefeller University, where he discovered adult neurogenesis in this species, also discovered the song system, and I think it was the location where echolocation was first discovered. So a lot of really interesting history of animal communication at Rockefeller University Field Research Center in Millbrook, New York. And at that time, I realized that you could record the entire repertoire of what a bird sings as he learns to sing, and that hadn't been done yet. And so the experiment that we did 25 years ago was to try to train birds with synthetic songs just by playing a tape to them. And we generated this in MATLAB, and these were songs that sounded like this. Okay, that's not going to work. Well, maybe I've got it on another. Okay, interesting. Well, it's not super critical for this talk, though it does add a lot. Let me see, maybe I can change my audio source here. I think it's about the latency. Yeah, it's... Let me see if I can do settings really quick. Mm-hmm. The DTV? No. Yeah. No. Okay. I think I can probably just do play from the computer. This will be found. This will be fine. It says output is black magic, but I'm just going to use the speakers here. So this will be fine. See if that works. Okay. Okay. What's that? Yeah, right? So that was the random walk through Canary Song in MATLAB. Thank you. Yeah, and these canaries would spend a lot of time working to imitate these songs. And this was one example of juvenile that after 6 months of practice came to imitate quite well. Oh, well, maybe on another slide. Let's see, this one, okay. So we had to be quite careful with that setup. The bird couldn't be around his father, so he was raised just with his mother. So, you know, we're certain that he hadn't heard another bird sing. If they were raised in that setting, they could be pushed incredibly far to imitate these very unnatural songs. Here, for example, was another synthetic tutor. Yeah. Couldn't get much simpler than that. And this is a canary after 6 months of practicing that song. 6 months? Yeah, it took a long time. So they work so hard to imitate these songs. Their whole life is just like organized around imitating that song. Okay. Okay, enough. The surprise, though, was that as the birds entered the breeding season, they reprogrammed those songs. So instead of continuing to maintain that imitation, they would produce a song that sounded like this. So they've taken those individual syllables from that imitated sequence and stuttered them in these repetitions. And that really surprised us because we understood that song was composed of genetic constraints, but also flexible imitation as a model of reinforcement learning. But we didn't realize the extent to which those constraints are really relaxed in the beginning, that they're actually really free reinforcement learners acoustically, and then the constraints can kind of come in as they enter the breeding season. And we wrote a paper with a depressing title that included, I think, the phrase, "Freedom in youth, rules in adulthood." And so that this is another example of that genetic organization underlying song. So if the birds were never exposed to another singer, and you just implanted testosterone in them, that 6-month learning process would accelerate into just 1 week, and they'd come up with actually quite a good song at the end of that 1 week, having never heard anything. So in my mind, this is like an amazing example of self-organization in systems neuroscience. It's something we really don't have great models for in AI, right? Like our AI systems don't self-organize. Reinforcement learning we have pretty good models for, but this is, like, I think, a really wonderful species to study this aspect of self-organization. Now, the songs are very complex. So this is, just an example of a typical song. They might have 30, 40 syllables, sing 60 seconds at a time when they sing. And if you go to great lengths to label all the syllables, they end up looking like big spaghettis when you build syntax models. You don't even see the light lines. I've just grayed out the lowest probable transitions here. So 20% of the syntax transitions aren't even highlighted here. They're just the gray lines. So it takes a lot of time to build these models, and my lab has been interested in what's the neural mechanisms of control of syntax, and if it takes 2 weeks or 3 weeks for a graduate student or undergrad to label a single bird song, that just really wasn't tractable to do neuroscience in this species. And this is one of the reasons that species like this just haven't really been studied at the level of neuronal control in any detail, even though they're really great models of the neuroscience of sequence generation. It's worth mentioning just some of the interesting observations that did come from manually labeled data sets. So one grad student back in 2013 realized that there are these quite long-range syntax rules in canary song. So here, if the bird sings this phrase that's orange and then pink, he'll follow with yellow, this syllable, if it was preceded by blue. But if it was preceded by this red sequence, it'll reliably follow with this purple sequence. So there are these long-range ordering rules in song that are quite interesting from a syntax perspective. We don't study meaning in vocalizations. Unfortunately, not yet. We don't try to translate. We're just trying to understand the neuroscience of the sequence control in the birdsong at this stage. But we are starting a project I'm not going to talk about, but we have a setup in the lab where birds can communicate with each other through iPads. And this records their video and their audio. And then we do the subtraction so we can get clean recordings from each individual this way and start to see how emotional state actually impacts the acoustic structure of their calls. And there's actually not a lot known about that yet in this species or really in any species. So to jump into the meat of the talk, this project wouldn't probably have happened without George Van Grovsky. He's a third-year grad student in the lab, very motivated to work on self-supervised learning and AI. And a former postdoc had successfully written some deep learning models to label song syntax in canaries just using LSTMs and supervised learning, straightforward. This we knew could be successful, but it still required a lot of training data on each individual to do that. It didn't really save us a whole lot of time. And so this project that I had failed at 25 years ago, can we do this in a fully automated way, is something that I pitched to George and we restarted. So the first kind of important thing to know, you guys are going to be familiar with this, spectrograms that are, even though it looks so easy, right? Our eyes can see these clusters. The spectrogram itself is not a good latent space or a good representational space for syllable clustering. And there was a question earlier about linear decoders as kind of a measure of the quality of a representation. So here we just quantify this by trying to train a linear decoder, so no nonlinearity is just a rotation matrix, to essentially classify the syllables and see if we can match the ground truth that the humans label just based on a linear transformation of the spectrogram itself. And that essentially fails, right? That the frame error rate hovers right near 100% frame error rate. And this is just quantified for 3 birds. These are human ground truths. And then this is UMAP representation of that spectrogram space colored by the syllables that we had in our ground truth data for the canary. You can get around this to some extent by not just trying to classify individual columns in your spectrogram, but open it up so you've got a little window, and then that's more data, more distinct. But There are downsides to this. It imposes certain time scales that are longer than the syllables. It doesn't allow you to be very sensitive to variable song, things like that. Just windowed image matching with like a dot product measure of similarity is not a very good approach. So we've talked today about a few different BERT models already. We focused on BERT as well, so it's very similar to GPT, but it's just a bidirectional transformer model. So the masked or missing data is something that you predict from both the past and the future. And first thing we did was look at aves. So this is the Earth Species Project model by Hajiwara. And it was just very clear that wasn't going to work for us. The temporal resolution is too low. So the time bin on that is 25 milliseconds, which works for humans, but not for the very fast syllables of Birdsong. And then the other thing which we didn't love about Hubert was that the tokenization involves this clustering step that's fairly complex. So there's k-means clustering of the audio, and then you iteratively refine that k-means as part of the training process. And we just found that to be somewhat complex. And so we wanted to see if we could do something with a faster temporal resolution, but then with a simpler front end that didn't tokenize the audio at all. So in a nutshell, we just developed a custom model from scratch that takes spectrograms. It masks out a portion of the spectrogram. As you can see here, it's essentially a pretty straightforward transformer model that then tries to predict the missing acoustic structure that was masked out. And then the strategy, as we've seen earlier today, is not to actually use that masked training criteria as the output of the model, but actually just as a pre-training step, and we look at the latent variables or internal states of that model to see if we get a better clustering than the spectrogram space for acoustic features. So during inference, we're not using that reconstruction, just generating these latent variables and then mapping that. Yeah, question?
Yeah, so if there's no tokenization, what's going into the model? Or like is there a vocabulary?
There's no vocabulary, it's just the spectrogram
it's like
itself.
A real valued.
Yep, continuous valued, I guess 0 to 255 kind of RGB, or 0 to 255 grayscale spectrogram, MEL spectrogram. Yeah. So yeah, no tokenization whatsoever.
Were you concerned with MEL at all? Because it's like for humans.
Actually, I think the next model I'm going to talk about was MEL. I think that we ended up using linear for this. So the first thing, which was kind of a curiosity here, well, let me first just say that this is essentially the structure. So it's standard convolution, linear projection, layer norm. There's I think 5 transformer blocks. The details are published on bioRxiv, and the GitHub is public. So all of this, you can get these details, but it's essentially a pretty straightforward modern transformer architecture. And one of the things that we did to say, are we just like in the right ballpark with the parameters for this model at all, might end up being useful in the future, and that's the following. We just initialized the transformer model with random parameters and then applied the same linear classifier and say, before we even start any training, can we actually classify the syllables better if we pass the data through the transformer first? And with certain parameter settings, we didn't do much to optimize this, but you can essentially see that the model with a completely untrained, randomly initialized transformer does far outperform the spectrogram in terms of classification. So this essentially is kind of support for something like a reservoir computing model of the transformer, that you're just using this as a way to mix the variables, and all by itself, before you even start training, you actually get an improvement in this classification. So I think that you could use this approach. This probably has been used for people that know this better. You could optimize the structure of the model using this prior to even initializing training. So I think it could be a really good savings. I imagine this is probably done, but something I'd like to explore further.
So that's random initialization. I'm curious if you tried to pre-train on something that isn't Birdsong, because there's like
didn't do that.
There's like this hypothesis that any training data will just imbue the model with some amount of structure that's already going to be really useful.
Yeah, we didn't look at that. I think that would be good. Okay, so training, I think it took about 15 hours of data per bird, 2.5 million parameters, so it's a pretty small model. We just trained on consumer GPUs in a few hours. NVIDIA 4096. And this is the kind of performance you get in that reconstruction task after pre-training. So these bars show the regions that have been masked in the data, and this is kind of the statistical structure of our masking routine. And then you can see that the model's doing a pretty good job of filling in the missing data after that masking. Here's just another example. This is, I think, a case where the model suggests that maybe there's another syllable here where in fact there's nothing, right? So there's some simple error modes here. This is actually a pretty rare type. So sometimes when the birds transition from one syllable to another, they produce an intermediate syllable type, and those can be quite variable. And so here you can see the model kind of fails to predict that because it's actually not probably seen any instance of that particular variable form. So this is how the linear classifier performs after pre-training. And you can see that just by solving that pre-training task, you end up generating latent space that's really useful. This is essentially a good indicator that the latent space maps onto the song in a meaningful way. And really, this is kind of our best case scenario. So if you actually open up all the parameters in the model, fine-tune it with our supervised data, you can ask, how well does that do? And that's the red state here. So the fully self-supervised model trained with where only the linear classifier is flexible for training does almost as well as opening up all parameters of this transformer. So this is what the intermediate states look like. So as the bird sings, these are the kind of latent variables in the layer that we were looking at. I think it was an attentional layer on the third or fourth transformer that we ended up focusing on. And then you can see the UMAP of these variables, and essentially you get these really nicely organized circles that correspond to each syllable. So for the most part in this data, a circle is a syllable, and the width of that circle corresponds to how variable that syllable is. So nice tight circles are very stereotyped syllables. These thicker ones are ones that might have a continuous variation in form, in essence, most of the syllables appear as ellipses, or in some cases like a figure 8 or a twisted ellipse, but essentially as ellipses in this 2D UMAP space. That was satisfying to me. Maybe not too surprising because the birds start, it has to be a closed loop because they're repeating themselves. And then, the simplest closed loop is a circle. It was particularly satisfying, though, because there have been mathematical models that I actually wrote during my PhD showing that if you try to reproduce birdsong from just simple mass spring models of the syrinx, you can do so with very good accuracy with just 2 dimensions of control, so pressure and tension, and essentially we showed that almost every syllable that the bird sings can be produced by an elliptical excursion in air pressure and tension in this one muscle in the syrinx. So if you build a biophysical model, the simplest biophysical model for the canary is essentially a circle in control space. And I don't want to make too much of it, but most of what the model discovers are circles, and that corresponds to the biophysics. So this is a comparison for 3 birds of the spectrogram embedding, if you just try to do direct spectrogram embedding, and then the self-supervised learning colored by ground truth. So these are colored by the human ground truth labels here. And here's an example of a single phrase with ground truth labels and then kind of machine discovered labels which we classify just based on a HDB scan clustering routine. So this is ground truth on the left, and then on the right is the clusters that would be labeled automatically with the HDB scan. And there's quite a good correspondence. So in fact, if you just say, what was the syllable that the machine, what was the label that the machine gives to a particular frame, and what's the label that a human gives, you essentially see this sparse matrix where there's almost a one-to-one relationship between what the machine labels as clusters and what humans label as clusters. With some exceptions where there's two clusters. The machine might label something as a single cluster where humans label it as two, for example. Here's one where the latent space is actually quite meaningful. The bird sings this syllable, and then he starts singing a second syllable with the other side of his syrinx. So he's got two sound sources, right? And so he continues to sing that same first syllable on one side of his syrinx, but then starts to activate the other syllable on the other side. And those are kind of two interlocking ellipses that humans label as just two completely distinct, at least the conventions in my lab were to label them as distinct syllables, but the machine essentially groups them as a single object with this kind of interlocking loop structure. Here's some examples of typical departures between humans and the machine. Human label on the bottom, machine label on the top. You can see there's like occasional errors. This is one where these really long syllables, the machine says, well, that's actually three different things. And I think actually that's a pretty good description of that behavior. The humans just grouped it into a single cluster. So can't really say that the machine does worse than the humans, and we'll get back to that. Yeah.
Yeah, on that. So the labels are just from this clustering, right? Mm-hmm. But I would think it's even fair to just take the embedding and then train, like I think you mentioned that earlier, train something from a few human examples and take that as your machine labels. Like, does that solve all your problems, or did it not change?
Yeah, we haven't tried to get the machine to correspond more closely to human labels yet by doing some kind of small number of clusters training. I think that would be a really good direction. Yeah.
It seems that the machine labeling is highlighting the points in which you can actually get out of the ring, right? So there must be something special there.
Yeah, this actually isn't well described by a ring, right? You'd actually need two rings to characterize it. Yeah. Yeah, I think that's exactly right. So now it becomes feasible to just automate. So this, instead of being a month of work, is just a few minutes of running inference. There's some subtleties on this, which is for 1 million data points, UMAP starts to become slow and unwieldy, and so we had to train another model to essentially reproduce the UMAP clusters. And if anyone has a good idea for essentially extracting low-dimensional states without going through a UMAP step, I think an end-to-end training that would actually give you low-dimensional bottlenecks comparable to UMAP would be really nice. We haven't succeeded at doing that yet, but a single end-to-end model that you could run without having to run the UMAP would be quite useful for inference. Okay, so you can say, well, just statistically, does the machine predict similar types of behaviors? And here's just a scatter plot of the phrase length for different individual syllable types from human labels and machine labeling. Here's another one for entropy or transition entropy, which is somewhat more variable. That one's very sensitive to any variation in the number of possible states. But the last one, and this is something that's kind of very new, but I'd like essentially to talk to you guys, hear some suggestions. What would be better ways of measuring the goodness of the machine labels? And I don't think that the human should be the ground truth here, because the machine is probably actually doing a better job at defining discrete clusters for this behavior than the grad students did. And just as one step in that direction, what we tried to do here was train a GPT-2 model to predict one sequence from another. And so this is training the GPT-2 to predict the ground truth labels from the ground truth labels. So it's essentially how well can you predict missing ground truth labels if you mask out some of them. And on the left is the validation loss. So the worst performer is actually ground truth human labels predicting ground truth human labels. The best performer is the machine label predicting itself, right? So that's actually, like, you can actually predict, the machine does a good job of predicting what it's going to label in the future. And then this was the one that was pretty interesting to me, the machine label prediction of the future human labels is actually better than the human labels predicting the future human labels. So if I give you, here's the history of everything that this human labeled, now predict what he's going to do on this next song, taking the machine labels for that vocalization, sequence of vocalizations actually did a better job of predicting what the human will do next than what that human, the history of what that human had done all the way up until that point. So, and so it basically tells me that if we can like formalize this, that might be a good way of somehow quantifying the goodness of this labeling sequence. First indications are that the machine is more consistent and can actually predict what the human does better than the human's own behavior predicts what the human will do next in terms of labeling. Okay. Validation of this, so the lab does do some biomedical research, and one of the areas we're really focused on is basal ganglia, and this is an area of the brain involved in stuttering, involved in Parkinson's disease, involved in Huntington's disease, and the canary, if we inject a little bit of an excitotoxic chemical in that part of the brain, the basal ganglia, in a canary, one of the grad students in the lab, Rose, has found that basically the birds start to stutter somewhat more aberrantly than they do to begin with. So there's a lot of stuttering in canary song, but they start to have these pretty long, fairly extreme forms of stuttering that occur after that basal ganglia partial lesion. And this just jumped out immediately from the model. So if you look at, pre-lesion songs and post-lesion songs, I think this is a sham lesion. So you can see that these are all the syllables the bird sings, and then just a plot of the phrase durations. Essentially, there's no change, but in the lesion, in one of these lesion birds, you can see that a few of these syllables becomes a huge variance in phrase duration, and it's just really satisfying that the model essentially does consistent labeling that's statistically really stable and can kind of pick up on these very discrete changes in the song post lesion. So this would have taken months of like human validation to label. 2,000 songs is many hundreds of thousands of syllables, and you can just do this now in a matter of minutes with inference. So that was our goal, and I think it's now available. It's something that we're happy to share, and I think a kind of a question for the group and for us is, the canary song's pretty special. The birds are really stuttering. There's not a lot of animal vocalizations that have that kind of periodicity, and to what extent will this model generalize to other maybe more complex, more challenging species remains to be seen. But one other question we're interested in was just, how special was that particular architecture in finding these good latent variables to describe canary song? And just as a preliminary, a student in a class that I taught this spring did a pilot study where we essentially took the masked autoencoder architecture, which is a transformer model that does a remarkably good job at producing images from massively masked data. So it's almost like, in my mind, unbelievable how well you can reconstruct, this image from this input, right? So this is a masked autoencoder model. So it's very similar in concept to what we've been doing with Tweetybird, but there's a kind of a distinct tokenization process where the data is kind of divided up into zones. This is a vision transformer model that's structurally somewhat distinct. I won't go into the details there, but what we did here, instead of training on canaries, we trained on Xenocanto. So this is the database of kind of user-submitted field recordings, and there were 500,000 field recordings in that data set. So we just trained a masked autoencoder on that. And here are some examples of how that pre-trained model performs on data from my lab after it was pre-trained on Xenocanto, but in I think we added the canary songs to the training set. We didn't add Bengalese finch song to the training set. But what you can see here is that now we're, instead of masking pretty small subsets of the data, we're essentially masking 75% of the data. You can see how incredibly broken it is, and that mask prediction is quite good. And in the case of a species that I think is mostly absent from that data set, at least our lab recordings weren't included in the pre-training data, it even does quite a good job of predicting the mask data for Bengalese finch song. And so we can construct a similar latent space from this masked autoencoder, and it's a little bit less beautiful in terms of these loops, but I think performs comparably. So I don't think that there's anything magical about the Tweety-Bird architecture. I think there's a whole family of architectures that are going to perform similarly on canary song. This one is interesting, though, because I think it really suggests that this essentially could operate as a foundation model where it's been trained with 500,000 species that are unrelated to canaries, and performs really well on the canary song without any fine-tuning on that species. Okay, so last thing I wanted to mention, and I have a small conflict of interest with this project, or complication of financial interest. I'm working as a consultant for the Swiss group, Signature, and they are producing these audio recording systems that are cell phone connected and integrated with a nice website that is able to quantify species that are found on this. And this is something that they're kind of still pre-fundraising on this project. So it's a project that's kind of in search of support. It's been hard to raise support for this through VC in Europe, but they have a bunch of VCs in Europe that are lined up and say, okay, we're ready to invest if you find an anchor. But this is, I think they have 600 or 700 pre-orders for this device. Maybe some of you guys know about it. I think they're planning to sell it for, I think, under $1,000, but it will stream and I think stays up for a couple weeks on the battery, and can also be obviously connected to solar. Cell phone connected. There's 3 integrated microphones, and very interested in also adding GPS, so you can do localization if you have put 3 of these in the forest and localize where these sound sources are coming from. I'm particularly interested in, and others in this room probably share this interest, but if we can go step towards identifying not just the species, but the individuals that are present on an acoustic monitoring station like this, I think it would just be amazing in the future to be able to say, this is George the song sparrow, and he was over in their yard, and here, that's where he spent his spring, and now he's back. And if we start tracking birds as individuals rather than as species, it just seems like that would do a tremendous amount for empathy and thinking of them as individuals. And so I think this is a really neat group. I have just really tremendous respect for them. They're both recent graduates from EPFL, and they're just very mission oriented on ecosystem measurement. And so if any of you guys are interested in this, I'm happy to put you in contact. All right, so closing with that, collaborations. Would love to work with data on whales and dolphins and coral reefs. So thank you. I'll take questions. Diana.
Thanks, Tim. That was beautiful. So with the birds you're studying, are the frequency and time domain, is it pretty stable and fixed?
Yeah, to some extent. So this next slide is actually an image of one of our birds through the spring and fall. And so what's quite interesting about canaries is they lose their feathers and they lose their minds in the fall. So they actually, like half of their neurons in the song system die and are regenerated. Mm-hmm. So the reptiles are constantly undergoing neurogenesis. Parrots don't seem to get dementia, right? So I mean, there's some things that humans could really benefit from in the biology of birds. But one of the things is instability. So from one year to the next, the bird song essentially falls apart and the bird is once again babbling in September. And so we trained a model for these guys that included both the spring and fall data. The fall data is very messy, so you no longer see quite the same clean loopy structure, but this is fall data and then that's spring data. So it's actually pretty stable during the breeding season, but then plastic throughout the year.
So thank you. So with marine mammals, for example, we've seen both experimentally and when we look in the wild that they can produce signals of the same contour, but they can vary greatly. Frequency domains and time domains and things like that. And I just was wondering how this would work, but you sort of are.
Yeah, I think you've touched on a really important point, and that is I'm kind of shocked that Tweety-BERT worked as well as it did, because there is quite a bit of variability. You know, some of the phrases have I guess these don't have the examples, but you saw the earlier examples where there's like some thick structure, there's like a continuous variation, or in those cases the birds are actually repeating a syllable where they begin at one frequency and end at a very different frequency, but the model kind of groups them all together into a single, into a single structure. I was surprised it was able to do that because our measure of similarity there is just pixel reconstruction of those spectrograms, right? And that's known to be super fragile for any kind of image transformer model, right? You don't need to get like perfect pixel reconstruction to get a good representation of, a school bus or something, right? So, so it was a pretty fragile measure of success that we built this model around, and yet it was still able to work. I think that for things that are more variable, like your dolphins potentially, I think we probably need to generalize to models that are doing things like making comparisons or reconstructions in latent space rather than in pixel space, and so there's the models like the JEPA models from there's that Meta. I don't remember who developed the JEPA models, but latent, essentially doing masking and then doing prediction in latent space rather than in pixel space, and much more complex to build those models, I think. But that would probably be where I'd go with more variable vocalizations.
Thank you.
Yeah. Here and then there. Yeah. Oh, yeah. You want to go first?
It's just a curiosity. I mean, is it possible that those fat circles that you have there really they just look like that because you projected down to 2D and they're really tori in the latent space, and that's actually what you're getting,
Possibly. I mean, definitely in some cases, the things that are quite far from circular are, probably a function of that reduced latent space. The HDB scan models do perform better in higher dimensions, so it's just the human projection in 2D. But yeah, if we actually want to generate labels, we'll project into like 6 dimensions cluster, and that actually looks quite a bit better.
Because it will give you more complex topologies on which to dynamics move, right?
Exactly.
It's not one ring extractor and then another, but like a composite, like a tensor component of them or something, composition.
Yeah. This was an experiment where I work in Eugene, Oregon, and we had to do this experiment, which is give birds psychedelics, and it's also like biomedically important and relevant, right? Plasticity, but this almost nothing. There wasn't a huge change here, but this particular syllable kind of developed this new structure here. So it's just a first indication that the latent spaces might be useful for picking this up. But, yeah.
All right. First of all, just in awe of like all of the visualizations. They're explaining everything so crisply. I know how much work it is. Every ablation and to get a visualization that's saying something could be weeks, and then you've just casually had so many of these in your talk. It's really incredible. And when we were actually building WAM, we were reading about Tweety-BERT and trying to figure out how to do things the same way. We ended up, we did a bunch of UMAP like a year ago, and we decided that it's too contorted and we actually don't have much to say about it. That's worth saying. So maybe one day. My question is pretty high level. At the beginning of the talk you said this is about syntax, it's not about semantics, and it sounds like that's the field of Birdsong analysis. We, or at least I, have a pretty clear holy grail of like semantics. Maybe step one would be to demarcate what is the meaning space that you can get from vocalizations. Where LLM is taking the first step, talking about dive and so on. Is that a direction at all that's interesting? And just my general ignorance, like what will be the function of all this sophisticated syntax that's predictable and nice, if not for semantics?
Yeah, it's I think the calls are much more rich in terms of syntax. So the birds have, these brief vocalizations that are much more variable, and I think that they correlate a lot with mood and emotional content than the structure of song. But what's known about the semantics, this really it's a breeding behavior, and the canaries. For example, some of these fastest syllables that they produce here, like this, involves coordination of the two sides of the syrinx, and they're like technically the most difficult. These aren't actually the fastest ones. They have some that are only 20 milliseconds long. So the shortest breath is 20 milliseconds in a canary, and so these are super, super complex syllables. You thought they must have worked like their whole life to imitate this from their fathers or mothers, and it turns out that those are the syllables that the females really like. So you can actually measure this in birds by putting estrogen in females, and they give the copulation solicitation display, and then it turns out they really like those syllables. Turns out those are also the ones that are just like genetically defined. So that like the bird will always invent those syllables straight from his genome without ever imitating. And so it's like a persistent puzzle of why is song so rich, right? So why are the birds of paradise so beautiful? It's just like kind of shockingly more than seems necessary for the task. In terms of semantics, I really think that I'm optimistic about this video setup that we've got in the lab, because we can, to some extent, reliably get birds to transition through different emotional states when they start engaging with each other through these video apps. So they, for example, you can give a bird a chance to have a Zoom call with himself with a time delay. And it turns out that they really don't like that. And so you can kind of see emotional state in the video kind of getting more, a little bit more anxious or perturbed. And then, by the end of it, they're like producing sounds that you've never heard them make with other birds. And so, I'd like to do the same for like inspiration and happiness, and not just go in the direction of annoyance in terms of studying semantics. But I think those calls are probably going to be a lot more rich for semantics.
So zooming with it itself is a setting, but zooming with others?
I'm sorry, the question?
Zooming with itself?
Yeah.
So that is you're replaying stuff that they've done in the past, right?
Yeah.
But zooming with it with other birds is okay? Because you said they're using...
They can get pretty agitated with that too, or interested, and it's very... I think it's a hard experiment to do, because they don't enter a steady state with respect to that. It's like what they do the first day they experience a Zoom call with another bird is very different than what they'll do the next day and the next day. And so, it's such an abnormal thing that, they're kind of going through this learning process, really essentially learning to ignore it, for the most part. But, by a week or two, they pretty much stop interacting with the Zoom calls. So, I think it'd be really nice if someone wanted to develop like models that actually tried to engage the birds through physical postures, and AI models that stand in as surrogates for another bird in those Zoom calls, and kind of create a reward function which optimizes engagement from the bird. And that might be an interesting way to kind of start to get traction on semantics too.
If other people have other questions,
Yeah, go
I'll
ahead.
hold this one till the end.
Mm-hmm.
I have a question about semantics, if we stay there for a while. And I talked to a lot of bird researchers, and the idea is that there's more to just the mating function. But like, do you have any speculations? Like you were mentioning emotional states. Do you have any other speculations of what kind of stuff semantically there might be?
I'm really not the right person to be asking this. We've focused on neuroscience of motor control and motor learning, and so this more, I think the semantic question is really new for us. So I'm happy to go down that path, but like the least qualified person in the room to speculate about that. I guess that the song quality must correlate with their, there's correlates with nutrition status, there's correlates with emotional status. So I think there's a lot of, just in the same way that the tone of our voice conveys a lot about emotional status or state. I think that's going to be a major part of the song, but beyond that, I don't have much speculation about the syntax of these complex songs in relation to meaning.
Like referential meaning.
Not for the songs. I just don't have a lot of maybe some others have some thoughts on
yeah,
this.
so this is the thing, right? Is like you often divide into songs and calls, and there's piles and piles and piles of observational knowledge about like alarm calls and begging calls and things like this, right? So the songs, which are often related to territory and mating, depending on who's receiving it, are sort of doing one set of functions, but then you've got a whole other pile of vocalizations for other sorts of functions.
Right.
I've talked to some folks that were studying like vocalizations. Yeah, and so another aspect of this is like what's actually available for us to observe. So songs are much easier for us to observe because they're putting so much energy into broadcasting those as far as they can. Mm-hmm. And then these short calls that are often shorter range, they're harder to observe. And as I was about to start saying, there was a researcher I was talking to in Australia who was doing like in-nest recordings and finding like the parent birds having like different vocalizations when they're showing up to the nest and when they're leaving the nest. So sort of a like hello versus goodbye kind of thing, maybe. So what do you have access to? What's going on?
And just to blur that.
Oh, sorry.
just to blur that boundary, there was a grad student in Fernando's lab that when I was a student there, he was studying begging calls in chipping sparrows and realized that the song actually evolves out of the begging calls. So that the begging calls get more and more variable, and then they kind of transition into babbling and into their song. So the foundation of their song is actually these begging calls. So I think, much like primate motor control or human motor control, there's a lot of these innate vocalizations that are essentially encoded at the level of the brainstem or at the thalamus, mostly brainstem, and they may be learned, but in the sense that they're fully self-organized. They don't require hearing to be able to develop most of these calls to first order, and then the song essentially comes into development later in the youth of these birds and innervates those same structures and starts to kind of play the keyboard. And so you've got this like frontal cortex that starts playing the keyboard of the calls, and then that evolves into this song behavior. So they're closely related evolutionarily, but semantics in song is just not something that the field has really pointed to, other than it's aggressive to sing the same song that another bird sings. So there's like, for a lot of species, that's like a super aggressive thing. So the bird talking to himself with a time delay, it was pretty clear that why that would have been frustrating to that bird. But beyond that, there's not a lot of semantic meaning in song that people have identified. So.
Last question, I'm curious. What kind of psychedelic it was and how much? There is a reason for this. But
yeah. So we used, in this case, DOI, which is available without DEA. It's essentially very similar to psilocybin, but it has a 24-hour half-life. And so it's not something that's generally abused by humans very often because it's like a week-long trip. And so it should have been like super potent, and there was like very minimal effect on the song of these.
So you didn't see like a larger reservoir of calls and everything?
Initial observation is nothing. And so this connects to another project in the lab where we are working with a group that has genetic factors that actually reopen plasticity, and so we can see, kind of compare those to the psychedelics.
That was the question, because in human studies that allows for plasticity to, for example, get out of mazings in which are depressive and so on.
Exactly. Yeah, I'm super interested in that. In human applications of neuroplastinogens, I think it's a very, very important area for future medicine. So, yeah.
All right. Well, that sums up our first day, and thank you very much.
Thank you, guys.
So we covered a lot of ground. We covered bats, corals, whales.